So when the very first next-gen sequencing data from metagenomic projects, namely marker gene projects, came out, we were immediately stunned by how inadequately prepared we were for handling those kinds of data sets. And I think back to, I guess it was December of 2005 when we saw our first examples of RDNA data sets from a pyrosequencing platform, and in those days we could only get 100,000 reads. Today we think that that's not very much at all. But I'll tell you, it broke. We were a molecular evolution lab, and it broke every single piece of software we owned. And so it took months to recover from that humbling experience. And so that ultimately, because of the efforts that we put in and the fact that we were working with a very large community of microbial oceanographers who were wanting to collect similar kinds of data, we decided that we needed to build some kind of an easy-to-use website or database that people could come and use without necessarily being super intelligent about writing Unix scripts or anything else. So in a sense, it was analogous to the Built Microbiome program, where you have architects and building engineers who don't have any reason to have an experience with handling molecular data. And so that gave rise to VAMPS. And so VAMPS is, I'm going to be talking about, Andy and Anya are here. They are the principal developers involved in the VAMPS initiative. Sue Hoos, she's the brains behind the entire program to speak of. I don't mean to embarrass you, Sue, but it is true. And my only job, and the only reason I'm giving this talk, is my job is to see if I can break the software. And I do that routinely. Okay, so this is VAMPS. I'll give you a website at the end. It's just designed to carry out a whole series of functions in terms of doing visual analysis of comparisons of ribosomal RNA databases in as simple fashion as possible, or easy to use fashion as possible. And this is a schematic that describes the way VAMPS operates. So VAMPS can operate in a sequence, in a taxonomy independent or a sequence independent fashion. And what I mean by that is you can work just on the basis of taxonomy, or you can work on the basis of operational taxonomic units or clustering of sequences. So this is really a common thread between VAMPS and QIIME. We just do it differently. In our case, we take data, initially we would take data from our output of our machines. As we got better at this, we started to generalize the capability of using VAMPS to users not using our machines, as you'll see as we go forward. And in our case, we take sequences. VAMPS does a lot of front-end processing that the user doesn't have to worry about. And that processing includes quality control, trimming sequences to what we would call an anchor. I assume that, Sue, you'll probably talk about anchor trimming some. And we essentially dereplicate sequences, and we do store them in a database that users can interact with on a sequence-by-sequence basis if they want to. So we end up with unique sequences, but we do bookkeep how many examples of each unique sequence there is in the database. And then you can either go into this split clustering routine and to form operational taxonomic units. It's just one of several different ways to identify OTUs. This is one that we think works and avoids some of the problems of sequencing error. And at least in the case of very short reads, it's very effective. The alternative is to use a process we call GAST, or you could use the RDP classifier if you prefer. And so you end up with a taxonomy labeled set of sequences. And into that, you can put it into a bunch of analysis tools where you can do either inter-community analysis or intra-community analysis. So intra is just pie charts and bar graphs and looking at sequence diversities, rarefaction curves, if you will. And I'll show you examples of that. I'll show you a few examples in inter-community analysis. And all the inter-community analyses are set up in a way that we think that you probably could convince us to use any R function, not any, but many of the R functions that are available to generate the comparison graphics that we provide. Okay, so this is what VAMPS looks like when you log in. So I just logged in. It doesn't quite log in that quickly. And by the way, I have to tell you that I thought about doing a real live demonstration of VAMPS in this room until I found how slow the wireless is. VAMPS is a problem in the sense that the way we're currently configured at the MBL, and I don't know if it's because of our pipe or what, is that it's very interactive with your machine. And so there's a lot of network traffic that does occur. So that's just a warning if you decide that 2 or 3 or 10 of you want to use it at the same time. You will run into trouble. Anyways, you log in, and now I'm obviously logged in, my favorite slide. You can get a VAMPS overview, and that VAMPS overview, I'm just screening it past this PowerPoint slide. It essentially summarizes the basic information I showed you in the first slide. Now that that's done, here I am back again. And I want to talk about, let's imagine you already have data uploaded into VAMPS, and we'll come back and talk about data uploads. One of the things we like to do is community visualization. And one of the features of VAMPS that makes it easier compared to the MG-RAST MobiDAC version is that we make it very easy to get out individual samples from different projects to do an analysis on. So these analyses are going to be done real time, that is, as you interact with the site itself. So it's not the case that you trigger off a process and you come back a half hour, an hour later. Most of the front-end processing that matters has already been done. So this is what you might see when we've gone to this visualization page. You'll see a list of different projects. And you'll also see, in the most simple case, you'll see a list of different taxonomic levels or even domains that you might want to compare data from. There is, by the way, a custom option that I'm not going to talk about because of time, but it allows you to work at mixed taxonomic levels. And if you want to know about that, I can briefly summarize that at the end of this talk. In any event, here are a whole series of projects. There are hundreds of projects now in VAMPS. We're just going to click on this one, and it doesn't look like this on the website, but I appreciate the fact that if you're sitting in the back of the room, you probably can't see half of these PowerPoints, so I've just expanded it a little bit. And this is just meant to show I'm looking at a project in Little Sipponisset Marsh, and it just shows that I can select different samples from Little Sipponisset Marsh, and I might analyze them at the same time as I might look at samples from the South Pacific Gyre, and I can select different data sets from different projects and mix and match them as I want to on the fly. One of the things that we have tried to do as best we can, and that is to collect metadata. We actually have very rich metadata sets from the International Census of Marine Life Initiative. And we have, if I understand correctly, I think we've turned off the entry of metadata into VAMPS right now. Is that correct? And the reason for that is that we're now forming this coalition with QIIME, and QIIME is going to take in metadata for us. And they have all these undergraduates under Gail's charge who go through and make sure the metadata is correct. And we found that metadata was the biggest headache we had with the International Census of Marine Microbes. You can't imagine how many different ways people screw up lat long, or how people use one set of units for the same measurement and someone uses a different set of measurements. And so we spent hours trying to do that, and I'm really glad, Rob, that you're going to be taking this, you have agreed to take this on, because we don't want to do it.
Yeah, we didn't really want to do it.
And there is some metadata in VAMPS. If you look at any of the KEC or the ICON projects, you would see a long list of metadata elements, but we're not going to spend time talking about that. We're going to now come back and look at an example of the different kinds of analyses once you've selected samples. And so you can do different visualization methods that range from looking at simple heat maps to taxonomy tables, and we'll look at examples of that, or bar graphs. Or you could look, and we won't be doing this, you could look at community dendrograms or alpha diversity plots, et cetera, et cetera. So this is all point and click stuff. You just have to be intelligent about the questions that you ask. Okay, so if we were operating at the phylum level, we might get a plot that looks like this. That's not very surprising. We're looking at a series of samples that really are not very related to each other. But at phylum level, they sort of look the same. Blue in our format are things that are the same. On the other hand, we could come in and change the taxonomic level to genus, and the moment you do that with this set of data, you see that the samples change very much in terms of the heat map propagation. So blue, once again, these are all South Pacific Gyre samples. they're all essentially the same thing. And then we see little syphilis Martian. And these are samples that are collected at different times, different stations, et cetera, et cetera. So there's some variation in terms of what their similarities and differences are. Other things you can do with VAMPS. You could do split heat maps. So you'll note that there are a series of different metrics that are used to compare the similarities and differences between these communities. I think, Andy, are you taking, these metrics are now being taken out of the R package at this point? Yeah. So you can pick any of these metrics, others could be added, and you can even generate split heat maps. And so in that case, you can now look in one half a domain of this heat map. What happens if you're looking, for example, at a Bray-Curtis measurement and compare that to what you see if you were looking at Jacquard just being presence or absence of taxon. So it's just that easy to get information. And if you don't like colors, you can ask it to give you values instead. So it'll give you decimal values as to how similar and different the communities are, and all that data is easily downloadable with a click. Let me go back to this. You could click on the intersection between two taxa, and it'll generate simple pie charts for you. And you can look at the pie charts in a variety of ways. One way is to look at it in terms of abundance, and so that's what we're looking at here. I know a lot of people look at pie charts, and they prefer to look at those pie charts according to the alphabetical array of the taxa involved. So you can choose as to which way you want to look at those, and you also get a listing. And so in this case, we're looking at a listing of the relative abundance of taxa, and of course we're at the top of this listing. The list goes on for a very long ways. And if you were to click on one of these pie segments, so if we were to click on this segment, it would actually go to the database and it would pull out the different sequences. And I know you can't see that in the back of the room, but you can't see it at the front. Well, would you like to borrow my glasses, Paula? Well, this is the problem with websites, is that projecting them, unless you want to zoom and expand them, is no good. So next time we have a meeting, Paula, if you would bring a wall of CRTs, that would be great. In any event, the most dominant sequence in that particular data set is this thing, which turns out to be a pseudo-ultra monad. And so this is the dominant sequence. It gives you information as to how well it was resolved. You can even drill deeper into this piece of data to find out how that taxonomic assignment was determined. We make use of a process called GAST. I was going to show you how GAST worked, but GAST has recently changed. It's a methodology that's more sensitive than just using BLAST to try and find the very best match against a reference database. I don't know if Sue's going to have time to talk about that. Probably not. Okay, so Sue's not going to go into it, and I took the one slide I had out of my presentation because it's no longer current. You can do other things. You can take that sequence and you could look for exact matches against any other sequence in our database, or you could blast our database. So you could look for any other community that might have that sequence, and it'll give you a report. Sadly, this takes some time. So that's one of the functions that's not quite so instantaneous. Here, we've come now back to looking at this two-dimensional, at this matrix, this heat map, if you will, and just want to show you that you can calculate PCOA plots pretty easily. And you could, so here's an example of the PCOA plots. You hit that button and you get some choices, so you can pick up different bits of metadata. You go ahead and do the calculation. Now, it doesn't do the calculation quite this fast, but it only takes a couple minutes. And you end up with some information. You can look at 2D plots, or you can look at the nice 3D plots that are very commonly seen when you make use of the QIIME program. Or you could download the mapping file, the data matrix file, principal component file, etc. And so this is just an example from that particular data set as to what two-dimensional PCOA plots might look like for absolute depth. This one, I think, was for salinity, and so on. Another feature of VAMPS, once you have your data in, you could ask it, instead of generating these heat maps, you could ask it, well, generate some bar charts. Bar charts are useful. And by the way, we can present these bar charts horizontally or vertically. We don't really care. It's your choice. But one of the nice things about these bar charts is that you have the ability to change what the relative where cutoffs are in terms of relative abundance. And so what I mean by that is you can see that there are some bars here that are very abundant compared to everything else, and if you prefer, you could take that number of 100% and you could drop it down to 10%, and now that changes the entire character of this bar chart. And if you wanted to just look at taxa that are really low in abundance, between 10 and 1%, you can just say go all the way down to 0%. And so now you start to see the very minor populations in a particular analysis. And so those kinds of tools are sometimes useful, and in fact, you can generate the heat maps, you can do many of the analyses that VAMPS does with selection of those dynamic ranges in terms of relative abundance. So if you think your communities are really interesting to look at in terms of low abundance taxa or moderately abundance taxa, you can constrain that. Because sometimes the most abundant taxa, if you look at water in Falmouth, Massachusetts, 90% of the reeds during the summertime will be Sphingomonadales. We can't see anything else. And we end up sequencing deeply enough, but the calculations become hostage to that overprinting of that one taxon. So it can become quite useful. And of course, you can generate this as pie charts. So all these things are very quick and easy to do. Okay, what about OTUs? Well, you can look at taxon independent OTUs, and we have a page for doing that. And this page has a lot of information. There are help files available to show you how you generate them. I'm not going to go through it. When you generate OTUs, that is a compute intense function, or it can be a compute intense function unless we pre-generated the OTUs for you. We've done that for all the V6 data we collect. We haven't done that yet for the V4, V6, or the V3, V5, or the longer reads, but we will be doing that shortly, I hope. So this is an example of looking at OTUs, not necessarily the same data set I was showing you before, but it's an example of OTUs that are now used in a heat map presentation. OTUs are, that is, these taxon independent OTUs are really useful because when we calculate them, we are calculating them against all the samples that we have in VAMPS of a particular sequence target. So we've OTU'd virtually all the V6 sequences in VAMPS. And so what that becomes useful is we could compare any two samples, and we could ask the question, what is the intersection in a pie chart format of all the OTUs in any two samples? So these pie charts for these two samples show you all the taxa that are in common. On the other hand, you can also look at all the taxon that are unique to either this sample or to this sample. So that kind of capability can sometimes be useful for you. And then another function I mentioned is you can generate these taxonomy tables, and the tables can be calculated in a number of different ways, but this is just an example of one of these tables, and you get out lists of the taxonomic assignments. You get numbers corresponding to how many times a particular taxon based on the GAST analysis appears in a particular sample. And you even have links to other sites, outlinks. So for example, we can go from this acetomic, so I'm not sure if it's going to be Ferritrix or microThrix that I did this with, but you can go to, for example, a wiki website. So it's acetobacteria, and you just click on that and you instantly get to a wiki website. It's amazing how quickly you'll start to learn microbiology by having access to that kind of a tool. I mean, a lot of us are molecular biologists and we don't know as much microbiology as we think we do. So web is a great thing. And then you could also click on one of these elements, and you can do this for a number of different taxa. It'll go ahead and generate plots for you across these particular taxa. You can also generate those plots as a function of some metadata element that you might select. So those are the easy functions available in VAMPS. So what about data uploading? Well, right now, VAMPS does have a pretty interesting, useful data upload capability, but the plan now in this this collaboration. It would be unequal partners if it were a marriage. I'm not collaborating. In this collaboration. So in this collaboration, the idea is that Doug and Anna, working with Rob, they've started to create a common portal for putting data into these sites. And this common portal is going to allow you to move data into QIIME along with metadata. And also you can choose to send the data to VAMPS. That's what the current plan is. The ultimate plan is, I think, that we will be exchanging data, which will be useful. So if you like some of the functions that we have in VAMPS, then you'll be able to take data from QIIME and put it into VAMPS. And if you like what we do in VAMPS and some of our data, the way we select taxon data, you can move it into QIIME. Just a matter of whether you like one versus the other, or you find one more useful than the other. Anyways, that's the plan. So this is what data upload looks like for us right now. So we're supporting, how do I do that? We're supporting, at the moment, we're supporting a number of different hypervariable regions, either in bacteria or archaea. We're supporting B9 sequences in eukaryotes. And then you have various parameters, and you can upload these data in a variety of ways that range from being FASTA or FASTQ, or I think SSF files are uploading if you can get the data to us. They tend to be larger. You have to provide some minimal comma-separated files, some CSV files with some information that we need. So we need information about the barcode, the run key. We need information about the name of the project, and there are some constraints in what we accept for names that you can read about. You can have a separate name for the data set. We want to know what the sequence direction is for your data. And we also have a project title. So this can be a longer title than what we have up here for each project. Project description, data set description, elements, environmental source of ID. The environmental source IDs are listed here, but I think we're going to see an expanded version of that significantly from QIIME. This is what a CSV file looks like. It's a pretty simple spreadsheet, not too difficult to fill out. We want to know what your primers look like in a CSV file so that we can properly trim the data sets. So once you have that information, you can load your data sets up into VAMPS, and we'll go ahead and process it. And, 8 or 9 hours later, maybe the next day, depending on how many people are attacking the database at one time, you'll have your data into VAMPS and be able to carry out most, if not all, the functions I just described for you, plus others. Others, examples of things that we do with QIIME routinely, shown here. Customized data sets. And what that means is sometimes people do experiments in triplicate or greater numbers, or sometimes they do experiments, they get exactly the same answer, but they would like to now combine this data into a single sample so you can increase your depth of sequencing. If you were, for example, looking for rare taxa at a given site. And so we have the ability to do that. So you could select some of these samples and you could form these custom data sets, which are really emerging of multiple sequence project, or multiple sequence samples coming together. So you increase your depth of sequencing, in essence. There are other capabilities. You can export data as FASTA files. You can export to cluster data. You can get out metadata. You can get out taxonomic counts, that is, row counts for each taxonomic identifier associated with a given data set, and so on and so forth. So there's a lot of flexibility, and you can be as dumb as I am when it comes to these kinds of Unix script-like manipulations and make good use of this system. If you want to know how to use VAMPS, there are frequently asked questions. There's also help pages that can give you access, although we need to spend more time updating some of this information. On the other hand, if you really feel lost, one of the things I'd like to point out is that we have this course at the MBL that we've now learned twice. Rob's group has contributed both TAs and Rob's time to this course, plus people throughout this community that are, not this community, but the bioinformatics community that are contributing towards the development of tools. And so it's a course if you have students. I don't have the exact dates in 2012, but it's just a 9 or 10 day course in which you become immersed on a 24-hour day basis working with various tools ranging from QIIME to VAMPS to Mother to other metagenomic data archiving and analysis tools. And so if you want to know more about VAMPS, that's its address, and I think you can extract from that information about how to get an account. And if you have questions, you can send mail to any of us on this list. And finally, I plan to make these slides available if you want on the microBEnet. You can make use of these slides. And I would encourage any of you who have spoken today, if you're willing to contribute your slides or sanitized versions of your slides, if there's data you're concerned about, it would be really nice to be able to put that information up that you've shown today.
Upload your slides to SlideShare and send me the link. That would be the easiest thing.
Okay, so I'm done.